Today we're going to be talking about the auditory system. Let's see, before I get started, I have a few announcements. One is the answer key to the problem set. My answers are up on Moodle. Just put them up this morning, so you should look them over. I hope to have them graded, yours graded by Thursday, but that's a little aspirational because of comps grading. So just, there's that in my background, not in your background. But I do hope to get them back to you on Thursday. Quiz 3 is this weekend. So just like the other two quizzes, you pick it up at the desk, you do it somewhere here, try to segregate yourself from people talking about it while you're taking it. That means you can go into another room, but it's closed book, so you put your stuff away or leave them at home when you come to take the quiz. If you see issues regarding academic integrity, you can either tell me or you can just post it to that anonymous thing, anonymous forum or whatever it's called on Moodle to let me know. You don't have to name names and you don't have to identify yourself. But I do want to know. PowerPoints and videos are up on Google, as always. Okay, so today we're talking about the auditory system. We are talking about hearing mostly, but we will also branch out or finish with the vestibular system. The auditory and vestibular systems are linked. They use the same kinds of receptors, some of the same pathways, the vestibulocochlear nerve, for example. So we'll talk first about hearing, anatomy of the ear, the map of the somatosensory modality, the sensory modality that hearing is, that is to say, tone. And we will go into molecular mechanisms of turning sound into signal that your brain can interpret. And we'll learn about some of the pathways from the basic nuclei of the brainstem to the auditory cortex, which is in the superior temporal lobe. That's the primary auditory cortex. And then we'll do some vestibular. So there's a video. The link is there. I'm going to go to it. And we're going to spend about 6 minutes getting an overview of the auditory system from this musical interlude.
The sense of hearing is accomplished by a process known as auditory transduction. The ear converts sound waves in the air into electrical impulses, which can be interpreted by the brain. As sound enters the ear, it passes through the external auditory canal, where it meets the tympanic membrane. The tympanic membrane then vibrates in response to the sound. Sounds of a lower pitch, or frequency, produce a slower rate of vibration. And sounds of lower volume, or amplitude, produce a less dramatic vibration. Higher frequency sounds produce faster vibrations. The tympanic membrane is cone-shaped and articulates with a chain of 3 bones called the auditory ossicles. They consist of the malleus, the incus, and the stapes. The movements of the tympanic membrane vibrate the ossicles, passing on the information of frequency and amplitude. The 3 bones pivot together on an axis shown here in red. The pivotal axis is due to a series of ligaments which hold the bones in place within the middle ear cavity. The anterior malleal ligament and the posterior incutal ligament are of particular importance for the pivotal axis. 2 structures which normally obscure this view of the middle ear have been removed. They are the chordae tympani nerve and the tendon of the tensor tympani muscle. Through the ossicles, the vibrations of the tympanic membrane are transferred to the footplate of the stapes. The stapes moves with a piston-like action, which sends vibrations into a structure called the bony labyrinth. The labyrinth is filled with a fluid called perilymph. If it were a completely closed and inflexible system, the movement of the stapes would be unable to displace the perilymph, and therefore unable to send vibrations into the bony structure. Due to the flexibility of a membrane called the round window, the stapes movement can displace the perilymph, allowing vibrations to enter the labyrinth. The corridor leading to the round window is found within the spiral portion of the bony labyrinth known as the cochlea. Vibrations produced by the stapes are drawn into the spiral system and return to meet the round window. The portion of the spiral passage in which vibrations ascend to the apex of the cochlea is called the scala vestibuli. The descending portion of the passage is called the scala tympani. A third structure, called the cochlear duct, is situated between the scala vestibuli and the scala tympani. The cochlear duct is filled with a fluid called endolymph, and when viewed in cross-section, the membranes separating the two fluid-filled systems are visible. They are Reissner's membrane and the basilar membrane. The membranes are flexible and move in response to the vibrations traveling up the scala vestibuli. The movements of the membranes then send vibrations back down to the scala tympani. A specialized structure called the organ of Corti is situated on the basilar membrane. As the basilar membrane vibrates, the organ of Corti is stimulated, which sends nerve impulses to the brain via the cochlear nerve. The actual nerve impulses are generated by specialized cells within the organ of Corti called hair cells. The hair cells are closely covered by a structure called the tectorial membrane. As the basilar membrane vibrates, the tiny clusters of hairs are bent against the tectorial membrane, triggering the hair cells to fire. The entire basilar membrane does not vibrate simultaneously. Instead, specific areas along the basilar membrane move variably in response to different frequencies of sound. Lower frequencies vibrate the basilar membrane closer to the apex of the cochlea, whereas higher frequencies produce vibrations closer to the base. This arrangement is known as tonotopic organization. Together, this sequence of events is responsible for our acoustic perception of the world around us.
Beethoven would be so excited. Now you know why you can hear all those annoying ads. So that's the overview. Let's get into some of the details. Some of them are correct in that. Some of them you won't need to know. Some of them you will need to know. Some of them I have to clarify. Okay, the outer ear. Let's start with the anatomy. The outer ear is called the pinna. This part of the outer ear is called the helix. Earlobe, you know that. The little thing in the front of the auditory canal is called the tragus. These might be extra credit, have extra credit value. Here's the auditory canal, the thing that goes to into your middle ear. So this is all the outer ear, auditory canal, and then the eardrum, the tympanic membrane. As the movie indicated, the eardrum is articulated with the three auditory ossicles, the malleus, incus, and stapes. I do want you to know the names of the auditory ossicles. I don't need you to know the names of those ligaments or muscles that the movie went into, that the video went into. The bony labyrinth consists of the cochlea and units of the vestibular system. So we're focusing today on the cochlea because we're focusing now on the cochlea because we're focusing on sound. But when we transition to understanding the vestibular system for balance, we will transition to this part of the bony labyrinth that has names that I'll get to when we get there. The stapes articulates into the bony labyrinth, and the fluid vibration, remember, sound is a vibration in the air. It's a pressure wave in the air that is converted by the eardrum into a movement of the ossicles, which is converted by the cochlea into a wave of vibration of the perilymph in the cochlea, in the scala vestibuli and the scala tympani of the cochlea. I'm just using the words that the video used. Amelia.
So like if you're in vacuum, can you hear when there's no
There's
air?
no sound in vacuum. So no, you can't hear because there is no sound.
Because there's no air.
There's no air. There's nothing to vibrate. So all those movies where you hear rocket ships whooshing in space, there's no sound. Not outside of the rocket ship. The cochlea is where the auditory hair cells are that are going to do the job of converting that vibration wave in the lymph, in the endolymph and perilymph of the cochlea, into electrical signals. The hair cells are the sensory cells of the auditory system. The hair cells are placed, the hair cells are going to be connected to afferent neurons of the cochlear branch of the vestibulocochlear cranial nerve. And the information is going to go to the brain. I could get ahead of myself, but I'm going to try not to. Questions about this level of anatomy? This is for the quiz. Yes?
Could you repeat what the phoenix was?
The pinna?
Or the one with an X?
Helix is just
Oh.
this part of the ear.
Oh,
It looks like a snail, sort of. Helix.
helix.
The pinna is the whole thing. Outer ear is an acceptable term. So this is the outer ear, this is the middle ear, and this is the inner ear. Question, yeah.
Could you go over the same
Go over the what? The bony labyrinth is the whole thing, the cochlea and the vestibular apparatus comprise the bony labyrinth. Yeah.
If your ears are like larger or smaller, does it affect how you hear, or
Maybe a little. Yeah, maybe a little. If your ears are out like this, you hear a little differently than if your ears are pinned back like this. A little. Not very much, but a little. Humans, I mean, compared to bats, where you have pretty crummy hearing, but compared to dogs, dogs can hear higher frequencies than we can. So our range is more limited than many animals, but we're not terrible on the animal spectrum. Pretty middle. Yeah.
What did you say? Was it the
Trochlea.
It started with a T on the
Tragus. Tragus is this little thing, some people have them pierced. Yeah.
What limits the frequencies you can hear physically?
The cochlea, the ability of the cochlea to vibrate at certain frequencies. So you'll see that in the book. The ability of the basilar membrane of the cochlea to vibrate at specific frequencies, to be more specific. The cochlea itself doesn't vibrate. It's the basilar membrane inside the cochlea that vibrates. Okay, so here is a enlargement of the cochlea, unraveled a little bit. So this is anatomically correct, but this is not. Okay, so the cochlea has been unraveled. But eardrum, malleus, incus, and stapes, perilymph in the scala vestibuli, movement of the stapes, vibration of the fluid in the scala vestibuli, to the apex of the cochlea. This is the snail apex of the cochlea. Then fluid, the vibration exits by vibration in the scala tympani, which is also filled with perilymph. And then the round window allows that pressure wave to have some place of exiting. Remember physics, fluid is not compressible. So if this was a hard structure, then this vibration wouldn't be able to move this fluid. There has to be an exit for fluids to vibrate. Inside, so scala vestibuli connected to the stapes. Scala tympani connected to the membrane of the round window. In between, fluid vibrates. In between them is, and this is enlarged out of proportion, is the cochlear duct, which is filled with a different fluid called endolymph. Perilymph, endolymph. Turns out that endolymph has a very specific composition, which I might get into. Perilymph is just normal extracellular fluid. Endolymph is a special extracellular fluid. Perilymph fills the scala vestibuli and the scala tympani of the cochlea. And it also fills what's not shown here, the vestibular apparatus. So here's some vibration transduction. The eardrum vibrates, the auditory ossicles vibrate, the stapes vibrates, vibration in the perilymph of the scala vestibuli, and then because of mapping of the frequency, a part of the, like if I played a single tone, a part of the basilar membrane is going to vibrate. Not the whole thing, just a part that is tuned to that frequency. So this vibration is going to sort of go up the vestibular, go up the scala vestibuli, get to a spot where it sort of resonates with the vibration. It gets to a spot where it then vibrates that part of the basilar membrane, and then the vibration wave exits on the scala tympani. And let me show you that a little bit better. Here. So here is a cartoon of the cochlea. Here's another cartoon of the cochlea somewhat unraveled. Everybody oriented with me? Eardrum, malleus incus stapes, the auditory ossicles, the oval window, the round window, scala vestibuli, scala tympani, and the basilar membrane between them. So, and here's the cartoon of it again. Scala tympani, scala vestibuli, scala tympani, cochlear duct with the basilar membrane in here somewhere. Oh, wait. Specific frequencies of sound vibrate specific parts of the basilar membrane of the cochlea. So this is tonotopic organization or tonotopic mapping. We map frequencies onto the cochlea. So the part of the cochlea that is closest to the oval window and the round window, the stapes and the round window, are specifically, the physics of it are such that the vibrations vibrate there best. And that's partly because the membrane at the base is stiff, and so it vibrates best at high frequencies. Low frequencies don't vibrate that membrane very well, so their vibrations penetrate further into the cochlea and vibrate, so very low frequencies vibrate the basilar membrane at the apex best. Now there's some vibration elsewhere, but this is the map. So high frequencies map to the apex of the cochlea. I'm sorry, the base of the cochlea, let me say that again. High frequencies map to the base of the cochlea, closest to the oval and the round windows. And low frequencies map to the apex of the cochlea, furthest away from the round window and oval window, because of the structure of the basilar membrane. The basilar membrane at the base is stiff. The basilar membrane at the apex is floppy. High frequency waves vibrate the base best. Low frequency waves vibrate the apex best, vibrate the basilar membrane of the apex best. So when I talk down here, I'm activating this part of your cochlea. When I talk way up here, I'm vibrating this part of your cochlea. And that range wasn't very good because, no, I did it completely backwards. Do that again. When I talk down here, and activating the base. No, the apex. Oh my God. When I talk down here, I'm vibrating the basilar membrane of the apex. When I talk up here, I'm vibrating the basilar membrane of the base. Did I get it right that time? And intermediate is intermediate. So we could play a game. Maybe I'll set this up at the break. Your hearing at high frequencies is better than mine because of age. So there are going to be frequencies that you hear that I just don't hear at all. And there are frequencies that dogs can hear that none of us can hear because their cochlea is different than ours. My hearing in the low range is probably just as good as yours. And that's because my basilar membrane has gotten too stiff to vibrate, or because the hair cells in here have been damaged by excess noise and vibration over the years. Going to concerts that were too loud. Yeah, playing earphone music. It's too loud. I've used my ears when I was younger. Yeah.
So what's the reason for some possibility of disease high frequency, very high pitch frequency?
Well, so, do you have a constant high pitch in your ear? No, well, I do. I have a disease called tinnitus. I am hearing a high-pitched screech in my ear all the time. It's annoying. It's actually in your brain. It's not in your ear. So let's wait on that. Yeah,
Is there a name for the white part of that?
the white part. The outside of the bony
Yeah.
labyrinth?
Is there,
The outside of the bony labyrinth, as far as I'm concerned. It's bony, it's hard. Okay, so here is a cross-section of the cochlea. So this is here's the apex, and here's the base. Scala vestibuli, scala tympani, cochlear duct, basilar membrane supporting the organ of Corti. The organ of Corti is where we're going here, because that's where sound is transduced into an electrical signal. So vibration, 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 movement of the organ of the basilar membrane, movement of the organ of Corti that is supported by the basilar membrane. So blowing up the basilar membrane in the organ of Corti, we have this structure here, the organ of Corti, and supported in the organ of Corti are the sensory transduction cells of the auditory system called hair cells. And the hairs are not hair. The hairs are little microvilli. No, yes, microvilli. So cilia, not hair. But they're called hair cells. They have nothing to do with hair. These are the sensory transduction cells. So these are the cells in the auditory system that the rods and cones are in the visual system. The cells that turn vibration into electrical signals. The video didn't go into that transduction at all, but you can imagine that that's where I am, that's what I think is interesting and exciting, important, so that's where we're going. There are other cells besides the hair cells that provide a lot of support, because it's a very well-defined structure, like the retina, and we're not going to get into that. There's some muscles, there's some support cells, we're not going to get into that. So, let's see the next slide. Okay, here's a cartoon of a pressure wave. So here are the hair cells, supported by other cells and sitting on the basilar membrane. So this whole thing is called the organ of Corti. The hair cells and support cells and the basilar membrane are components of the organ of Corti. When the organ of Corti when the basilar membrane is deflected, the hair cells move a little bit because of that deflection. And because the hairs of the hair cells, this is where we're going, the cilia and the microvilli sitting at the top of the hair cell are embedded in this other structure called the tectorial membrane. Movement of the basilar membrane, movement of the cells that are supported by the basilar membrane, less movement of the tectorial membrane causes bending of the microvilli and cilia of the hair cell. That bending is going to be critical. So here's a cartoon. Yes, question?
So the movement causes less bending, is what you said?
No, the movement causes more bending.
Okay.
Well, the movement causes more and less bending because it's a vibration. So when it moves up, it causes more bending. When it moves down, it causes less bending. I do want you to know the names of the hair cells, but I don't care that you discriminate between inner and outer. That's a distinction that isn't necessary for this course. If you become an audiologist, which is a career out there, if you're interested, then you will obviously learn more about the mechanism of hearing. So yeah, I never pitch audiology, but it's a career. I have seen audiologists because of the tinnitus that I suffer from, and they're interesting people. They know a lot of medicine, and they can be very helpful for people with hearing issues, including hearing aids or other devices to assist in hearing, etc. I think most audiologists now is a master's in audiology. I don't think it's a doctoral, but there could be. There's certainly PhDs in the neuroscience of hearing, but if you're interested in the clinical aspects of hearing, look into audiology. So here's a cartoon of the hair cell. It's a simple cartoon. It's got a nucleus. Yeah, they give. What else does it have? It has endoplasmic reticulum. It's certainly got mitochondria. It's got all kinds of that good stuff that you learned in self-is, and a nucleus that you learned about in CG. Here is the bundle of quote-unquote hairs, but they're not hair. They are microvilli. They are extensions of this membrane. They're membrane-lined. Hair is not a membrane. Hair is keratin. This is a extension of the membrane, and they form this well-defined bundle leading up to a structure called the kinocilium. We'll get to that in a minute. This is the hair cell. Movement of the hairs is going to be turned into release of neurotransmitter from the hair cell, and that's going to activate the afferent of the nerve that is going to go to the brain. So is this a long or a short receptor cell?
Short.
It's a short receptor cell. The hair cell, the receptor, does not have an axon. It's connected to a neuron that has an axon. Do you think hair cells fire action potentials? No, this is where the movie was wrong. The hair cells do not fire action potentials. They release neurotransmitter. So they have voltage-gated calcium channels to trigger calcium that does all that stuff with snares and synaptotagmin to release neurotransmitter, but they don't fire action potentials. The hairs, the quote-unquote hairs of the hair cell, the cilia and the microvilli of the hair cells, are embedded in the endolymph of the cochlear duct. Remember, this is the scala, this tympani. Let's see, let's get oriented. Oops. This is the scala vestibuli. This is the endolymph. Here is the organ of Corti. Scala vestibuli, endolymph. Endolymph surrounding the surface of the hair cell, where the hairs are. Remember, it's not here. Here are electron micrographs of hair cell bundles. So this cartoon is not fanciful. They're beautiful cells. They're incredible. So here is a hair cell bundle in somewhat three dimensions. The microvilli are organized short, medium, and long short, medium, and long, leaning up against a structure behind it that supports the microvilli. So this whole thing is the surface of this hair cell that contains this hair cell, this hair bundle of microvilli and stereocilia. Here's some more pictures of electron micrographs of a organ of Corti with the bundles of hair cells. So now we're going to get into transduction. Molecular mechanisms that convert the bending of the microvilli and stereocilia into a electrical signal, which is then going to be turned into release of neurotransmitter. Are there any questions? This is a good spot for questions. Yeah, so
Is it just one neurotransmitter released, or just?
so the neurotransmitter that is released here is still somewhat debatable. There's uncertainty of the neurotransmitter here, at least the last time I read the literature. So I'll admit, I didn't read it this morning, or even this year, 2020. But I won't ask you to memorize the name of this neurotransmitter because I'm not convinced we know it. Remember, the first neurotransmitter in the retina is glutamate. That's known. The nature of this neurotransmission is less well known, at least by me. Somebody might feel that they have it, they know it, but I'm not sure.
So how do they just?
They know it's
like,
excitatory.
How do they know like which frequency it is with the neurotransmitter? Just where on the membrane?
Where on the membrane?
Okay.
Where this hair cell is on the vascular membrane is which hair cells are vibrating, which is
Which neurons go to?
which ones are releasing neurotransmitters, which nerves are taking information to the brain. Yeah.
Maybe we're gonna get into this, but like, is there a physiological relevance to the fact that some of the hair cells are long and they like?
Yes, we're going to see that. We're going to see a relevance to this stacking of hair cells right now. It is relevant. So here's the relevance of it. Let's start with a hair cell bundle that is bent, with the short ones bent away from the long ones. Right? We're starting at this point. With the short ones bent away from the long ones. There is a structure that couples the tip of the short one to the side of the longer one that it's next to. And it's called the tip link. Some people call it the gating spring. It's a protein. It's an extracellular protein that is anchored at the tip of the short one and anchored at the wall of the long one. When this structure, so here's a short one, here's a long one. Short, long. When this structure is bent towards the short one, the distance between the tip of the short one and the point of attachment of the long one is that distance is shorter, and so the tip link is not stretched. That not stretch, it calls it compressed, but it's just not stretched. That not stretch of the tip link allows cation permeable channels on the tip of the shorter one to close. These are stretch activated cation channels. They have just been characterized. They got a molecular name. I forget the name of it. It's a year ago when I said this, I would have said, we don't know the molecular composition of this stretch activated channel. We do know it now. It's a bunch of letters. I'm not asking you to learn it, but I could tell you about the molecular structure of this stretch activated cation channel here at the tip of the shorter stereocilia. So when the stereocilia are bent towards the shorter one, the tip link is not stretched. The stretch activated cation channels are not open. There is not influx of sodium and calcium. So the membrane is polarized or hyperpolarized. Less neurotransmitter release. You know why. Less action potentials in the nerve that leaves the organ of Corti. When the tip link, when the hair cell moves, bent in the other direction, the distance between the tip and the tip of the shorter one, and the wall of the longer one, that distance increases. That stretches the tip link. That opens the stretch-gated cation channel, causes a depolarization, opening of calcium channels, voltage-gated calcium channels, calcium influx, synaptotagmin, snare complex, neurotransmitter release, excitatory neurotransmitter release, increase in the frequency of action potentials in that auditory nerve. So here is you might not believe me, you'd be justified in thinking, what, this tip link must be invisible, it's so small. Well, it's almost invisible, but electron microscopy shows this structure coupling the tip of the shorter one to the wall of the longer one. So in a bundle like this, there are tip links that we can't see here, connecting the shorter one to the wall of the longer one. And when this tip bundle moves, let's say moves towards us, when this bundle moves towards us, the tip link is not stretched. And when this bundle moves towards the back, they're all stretched. A bunch of stretch activated ion channels, depolarization of this hair cell, release of neurotransmitter. Joey?
So can there be multiple tip links like on a certain hair? So like since the one like in the middle would be like technically the long one, but then it would be the shorter one for the one behind.
That's right.
it.
So this is the shorter one for this. So this tip link array here is this short one connected to this next longer one.
Okay.
And then there's a tip link from here to the wall of here. And there's a tip link from here to the wall of here. Yeah, there's this. Did that answer your question?
Yeah.
Yeah. Uh-huh.
What determines which way it bends?
Which it bends back and forth. And which ones are bending depends on the frequency of sound and where this hair cell is on the basilar membrane. And when I said there is a tip link, I mean there's a tip link from here to here, there's a tip link from here to here. All of these microvilli, all of them, each of them, has a tip link between the tip and the wall of the next larger one. And the only ones that don't are the tallest ones, because they're bending up against a special structure that doesn't move really. Well, moves, but it's not a transduction mechanism. Yeah.
Are they ever stretched in first?
At the same time? No. So this is the, yes?
So is the baseline no movement or is the baseline stretched?
I think the baseline is in between, and you would say that that's not stretched or compressed. So this is transduction. We've transduced of air vibration. Let's review. We've transduced a vibration in the air into movement of some bones, the auditory ossicles, but because of the eardrum. We've turned that movement of bones into movement of fluid. We've turned that movement of fluid into a vibration of a membrane. We've turned that vibration of membrane into bending of the hair cell bundle. And the bending and unbending of the hair cell bundle has been converted into depolarization or hyperpolarization, which causes bursts of action potentials, a burst and then no burst, a burst and then no burst, depending on, so burst, no burst, burst, no burst, burst, no burst. Which nerve is carrying that burst, that information, that bursting of action potentials? Which nerve is carrying it is what carries the pitch information to our brain. Because which nerve carries it is determined by where on the cochlea that hair cell that we're talking about is located. So when frequencies of, let's say, 5,000 hertz, if I played a tone of 5 kilohertz here, there'd be a part of the membrane that is bending specifically, a range, and those hair cells are sending bursts of action potentials to the brain, and these hair cells are not, or sending fewer bursts. When I play a low frequency sound, these hair cells connected to their nerves are sending bursts of action potentials to the brain. And not these things. Okay? Let's take a break now, and we'll talk about the information, the bursts of action potentials, and where it goes, and what we do with that information. Let me just clarify a little bit. I think I got the names scala tympani and scala vestibuli mixed up when I was talking about this slide. Just to review, vibration of the eardrum, vibration of the ossicles, vibration of the stapes, vibration of the fluid in the scala vestibuli. Because of the specific organization of the cochlea, there is a spot on the scala vestibuli, where that vibration vibrates maximally. It's kind of a resonance sort of thing. So there's a spot where this is vibrating maximally on the basilar membrane. That moves the basilar membrane, and then the vibration exits the scala tympani to the round window. The movement of the basilar membrane and the bending of the hair cells there is what tells us that there's sound, and the location of that on the basilar membrane is what tells us whether it's a high frequency or a low frequency sound. High frequency, low frequency. So which nerves from the cochlea carrying bursts of action potentials is what carries information about the frequency of the sound that you're hearing. The nerves exiting the vestibulocochlear nerve, or nerves of the cochlear branch of the vestibulocochlear nerve, make synapses in the medulla. I'm not going to ask you to learn the names of the nuclei here. So where is the first synapse of the auditory pathway? It's in the cochlea, because This is a synapse. The first synapse of the auditory pathway is in the cochlea. Just like the first synapse of the pain pathway is in the dorsal horn. And the first synapse of the visual pathway is in the retina. Knowing that organization is part of knowing the anatomy. The next synapse is in the medulla. That's really all you need to know. And then it gets complicated. And it goes through various midbrain and thalamic processing. So medulla, midbrain, thalamic. Some of it, most of it is contralateral, but not all of it, so we don't have to worry about contralateral, ipsilateral in this setting. And it ends up in the auditory cortex of both sides, but let's just say both sides. The auditory cortex is this superior temporal gyrus. So what do you need to know from a slide like this? Well, I think I just walked through it. First synapse in the cochlea, next synapse in the midbrain, process in the medulla, processing in the midbrain and the thalamus. Unlike in the visual system, I asked you to learn the name lateral geniculate nucleus. In this case, I'm not really going to ask you to learn the name medial, but if you know the name lateral, and I say, what does the medial geniculate nucleus do? You know it's not part of the visual system, so you might guess, and you'd be guessing correctly, it's part of the auditory system. And then neurons project to and make synapses in the primary auditory cortex, which is in the superior temporal lobe, the superior temporal gyrus. Question? Yeah.
On that previous slide, is that a sagittal section of the brain?
This, you mean here?
Yeah.
No, that's a horizontal section of the brain.
Oh, right.
So here is the midline, this is the longitudinal fissure here. These are the two lateral ventricles on either side of the longitudinal fissure. This is anterior, this is posterior. So this is a horizontal, we don't do this very much. This is a horizontal section through the brain showing the superior temporal gyrus on both sides. Yeah.
Do we need to know like where the crossover happens?
No, not in this
Just-
pathway.
Just that it's contralateral?
Well, it's not even purely contralateral, so you don't have to know that. Some of it ends up contralateral, but unlike in the pain pathway where it all goes contralateral, you don't have to know that here in the auditory pathway. So, yeah, this is confusing. This is a coronal section showing the auditory cortex on the left side and the auditory cortex on the right side. This is a horizontal section showing the auditory cortex on the left side and the auditory cortex on the right side. Are we clear on the orientations of what we're seeing here? So the primary auditory cortex has a map, right? We talked about the map of the somatosensory cortex, where the body is mapped on the post-central gyrus. We talked about the map of the visual system, where the fovea is mapped most posterior in the gyri above and below the calcarine sulcus, the most posterior part of the occipital lobe. That's the visual map. We have an auditory map where we map frequency, because that is that's a lot of what we're processing. It's the pitch. So here's a lateral view, anterior, posterior. temporal lobe, anterior, posterior. Low frequencies are mapped anterior, high frequencies are mapped posterior. That's what I want you to know. As we go more lateral, we're not, so going this way, so this is low frequency sound is being processed in this part of the primary auditory cortex. High frequency sound is mapped in this part of the primary auditory cortex. Which ear it's coming from is mapped laterally, and that's what I don't want you to know. So there's, in addition to processing sound, tone, that is to say frequency, tone, not the high pitch or low pitch, we process where the sound is coming from. That's why we have two ears on either side of our head. There are two mechanisms for localization of sound in the brain. One is that our brain can tell that the sound is coming from one side of the head sooner than from the other side of the head. So a sound coming from over here is going to activate this ear, activate all the systems that I've been telling you about on this side, about 20 microseconds before it's going to hit this side because of the speed of sound and the distance between my two ears. So we detect this delay. We have a beautiful mechanism for detecting this delay in the medial superior olive. This is the nucleus that I didn't ask you to learn about, the olivary nuclei of the medulla. We have a beautiful mechanism for doing this. And it just has to do with the medulla being in the middle, and this nucleus being in the middle of the medulla, and the axons from one ear are closer to one side than the axons from the other ear. So let's just take an example where the sound is coming straight in front of me. It's activating neurons from one ear and activating neurons from the other ear. And the part of the medulla, the part of the nucleus that is getting activated maximally at the same time by both ears is right here. This ear is getting this sound just a little bit before it's getting this sound. And so it's not being maximally activated when it's straight in front of me. If it's coming from my left side, this part of the medulla is getting activated early. And this part of the medulla is getting activated late because it's longer, and that maximally activates this part of the medulla. I'm just teaching you this because I think it's so cool, that it's the length of the axon that helps us determine where the sound is coming from. I will not ask you to reproduce this mechanism on an exam. The point is that simultaneity is what maximally activates part of the medulla, which simultaneity activates which part of the medulla? Which tells the brain it's coming from a location. So let's play a little game. We're pretty good at this. We're pretty good at this. So I need a volunteer. Joey, come on up here. Stand over here. Trying to get away from that noisemaker over there. So stand right here. Close your eyes. Oh, I have to give you instructions. Point to where you hear the sound. I'm going to be quiet. I'm just going to click my fingers. And you point to where you hear the sound. Okay? With your eyes closed. Getting it almost, I mean, he's not 100% accurate, but he's really close. That's good, thank you very much. So he's detecting that by, oh, one more thing. So, go back. So this is how we detect position in this orientation. But you might realize, wait a second, a sound straight here and a sound up here is going to sound the same by this mechanism. But you know we can detect the third dimension, and that's what we're going to get to next. So the third dimension is detected by modification of the sound by our outer ear. We learn how to process. It's basically signal processing, and the outer ear changes very subtly the nature of the sound. So let's do this again. Point to where the sound is coming from. So even though this sound is hitting his ears at the same time as this sound, I mean, the difference between the sound is, this is a sound that hits our two ears simultaneously, and this is a sound that hits our two ears simultaneously. He can tell the difference between this. Point. Between this and this. That's what I mean. Thank you. So we use those two mechanisms to localize sound in our three-dimensional world. The timing of the hitting of the two ears and the processing of the sound in the third vertical dimension. Let's play a quick, let's see if I can find, I forgot to dig up one of my favorite websites. I don't want to hurt your eardrum, so I'm going to turn the sound down. Just to make sure I don't hurt anybody. You can hear that sound, right? Question?
No.
Okay. Yeah, we're all good at hearing this medial tone. Okay, I can hear this tone. Tell me when you raise your hand when you can't hear the tone anymore. Is that hurt? I can't hear a thing. You can hear something now?
Yeah.
Sorry. I couldn't hear a thing. And I can't hear it either. The sound system doesn't have perfect linearity across the frequency. So some of this is the ability of that speaker to transduce this vibration, and some of it is our eardrum, or ear able to hear that sound. So this is not a perfect test because the sound system is not good, but you can tell that high frequency sounds Where are we hearing this? Where on our cochlea is being vibrated? Closer to the base. And where is this vibrating? Closer to the apex. And we can discriminate those tones. That's what I meant. Oops, that's not good. Okay, let's switch gears. Question?
I was wondering why like ear damage only affects a certain frequency.
So, certain parts of your basilar membrane are damaged. So in my case, I probably damaged the part closer to my base. So those hair cells are not sending a signal to my brain anymore. And my brain grew up knowing there should be tone in there. Why am I not hearing anything in that region? I'm gonna make something sound like that. And I'm filling in the gap. So my brain is saying, there should be some sound in there, let's make some sound in there. And it's not sound, but it's in my head, a screech. High-pitched, metallic tone.
Is it because the base is so close to the stapes? And that's why it gets...
I don't know why parts of the ear, I mean, so in principle, I suppose you could get damage anywhere, depending on what frequencies. So if you were in an accident or, a bomb blast, and a specific frequency really hit part of your ear, I suppose a low frequency sound could get, those hair cells are pretty fragile. And they don't regenerate. Yeah.
So if you burst your eardrum, is it because it was vibrating too much?
Yeah. Yeah, but the eardrum can regenerate. Okay, vestibular system. So we've been here, now we're moving to this part of the bony labyrinth. The mechanism for transducing movement of our head is the same. It's not in the cochlea, it's in the cupula of the ampulla. So this structure is the ampulla. Inside the ampulla is the cupula. That's where the hair cells are. There is perilymph filling these structures. So we have 3 semicircular canals that are oriented in 3 dimensions. It's hard to tell here, but this semicircular, it's a circle, but it looks like an oval because it's mostly coming out towards us. So this is a circle, this is a circle, this is a circle, 3 dimensions. And so when I move my head in this dimension, I am moving fluid in this semicircular canal. And when I move my head in this dimension, I'm moving fluid in this semicircular canal because of the, inertia of the fluid, right? I move my head, the fluid moves, but it lags behind because of the inertia of the fluid. So the semicircular canals are geared towards understanding rotational movement of the head. In 3 dimensions. Movement of the fluid is going to cause bending of hairs in the cupula, and that's going to be transmitted to my brain as a signal, oh, your head is moving. And this is why you get dizzy. If I do this, I'm not trying to follow it, but if I do this, I'm getting the fluid in my horizontal semicircular canal moving. And so now it thinks I'm still moving, but my visual system says, no, you're not moving, and I'm really dizzy because I'm getting 2 conflicting signals from the vestibular system and from my visual system. My auditory system, my vestibular system is saying, you're still moving in the other direction because the fluid was moving. I'm getting better. The fluid was moving, but my visual system says, no, you're not moving, and that's dizziness. Okay? So if I do this, I don't get dizzy because I'm just moving the fluid back and forth. The fluid moves the cupula supports hair cells, and the bending of those hair cells is transmitted to the brain as a signal that says your head is rotating. So the semicircular canal's rotational movement, the cupula inside the ampulla at one end of the canal is where the transduction happens. These other structures, the saccule and the utricle, these are all names I do want you to know. The saccule and the utricle detect linear. They are not detecting rotational movement, they're detecting linear movement. So when I move my head this way, or when I move my head this way, I'm activating, or when I move my head this way, linear movement in any 3 dimensions, I'm activating hair cells in the utricle and saccule. Hair cells in a structure inside the utricle and saccule. So rotational movement, semicircular canals. Linear movement, utricle and saccule. Hair cells, action potential, same idea. Hair cells, neurotransmitter release, action potentials in the afferent neuron. The afferent neuron of the vestibular branch merges with the axons of the cochlear branch to form the vestibulocochlear nerve. Which axons are carrying action potentials tells you whether your head is rotating or moving this way or moving this way. Which axons carries which type of movement modality. This is not on your handout, so you don't need to know the details, but the point is the first synapse of the vestibular system is in the vestibular apparatus, because there's a synapse right here between the hair cells and the afferent neuron. Right, there's a synapse. The second synapse is in the pons, and the information from the vestibular apparatus is mostly going to the cerebellum. It is not going to the auditory cortex. And the cerebellum is critical for us. When we get to the motor system, we're going to see the motor system, the cerebellum is important for fine motor control and balance. So, the fact that I can do this, sort of, is my vestibular system and my motor system working together to do balance. Which muscles do I need to use to do some balancing act like that? Vestibular apparatus of the bony labyrinth, pons, cerebellum. So that's the level of detail I will want you to know. Just like I will want you to know auditory system, medulla, midbrain, thalamus, auditory cortex. Okay, we're good for today. I'll see some of you this afternoon. Others, I'll see